Hello, Science 30. Welcome to protein synthesis. We are going to discuss how your DNA ends up making all the proteins that make up you. So when it comes to the compositions of proteins, we're going to talk about where they are, what they are, and how they're made. But for the entire portion of this video, anytime I talk about something called an amino acid or a codon or a triplet, you're going to refer to page 13 of your data book, which has your amino acid chart. So your body, which is made up of 20 different amino acids. So only 20. You would think there's a lot more, but there isn't. Each of these acids or amino acid sequence end up identifying a protein. We call these polypeptide chains. Now, amino acids, when they're put into certain orders, they end up making different types of proteins. So what we do is we sequence the bases of the DNA to determine the sequence of the amino acids. Essentially, what that means is we read the DNA, and the DNA tells us what amino acids we need to make for certain proteins. So for example, your eye color is a certain color made out of protein, okay? If you have a certain section of your DNA that codes for your eye color, you would read that, then you would make proteins out of it. Every gene is equal to one protein, and the polypeptide chain, that long chain in the picture there, is made up of a bunch of amino acids linked together by something we call peptide bonds. So if you look here in the picture, you can see phenylalanine, leucine, serine, and cysteine, and you can see these little black lines that attach them. Those are represented of polypeptide bonds, okay? And then this long, colorful chain right here is known as a polypeptide chain, and it'll fold up and make a protein. So this picture right here is representing your data chart in your data booklet on page 13. So what you can see here is this is called the DNA-based triplets and their corresponding amino acids. So how you read this chart is on the side here you have the first base, along the top part you have the second base, and then along this side you have the third base. So as we go through, I'll show you what that looks like. So each amino acid is coded for by 3 nitrogen bases on a DNA molecule. So anything that is an A, T, C, or G. Okay, so A, T, C's, or G's make up DNA, which we know makes up eventually a protein. Each 3 nitrogen base is called a triplet. So you might hear me use the words triplets or codons from now on. I'm just referring to 3 letters like TAT or TCC. That is called a triplet. In a DNA triplet, AAA, we code for the amino acid lysine. How we know this is, again, we read it first base, second base, and then third. Here is A. So I follow A along the top looking for the A's. Okay, so here's the A base. Now I go over here and I say A here, and here it is, lysine. So A to A to A. Here's lysine. That's how you read it. We are going to be using this data booklet anytime I ask you to look up a DNA triplet. So anytime I say find the amino acid, you need to use this chart. So we're going to look for and code these following triplets. So you have these in your notes. What I would recommend is pausing, trying it out on your own, and then looking at my example. So TGT, how do we read that? Well, T's here, T's here, G's over here, so I'm going to follow along. And right here, TGT. I said TTG. So TGT. So we have cysteine. C, A, follow it down to T. We have histidine. A, and then to T, which is right here, then to G. We call this the start codon. It starts almost every protein chain we have. And if it's in the middle of a protein chain, we call it methylamine. And last but not least is T, G, and then we find A. And this is one of the 3 stop codons that we have present in our chart. So you'll notice at the bottom of this chart, there's a couple notes. It says, note, this table is used for bases of triplets from the complementary strand of DNA. So complementary strands is 5 to 3. 5' to 3', we call it the coding strand, and I'll explain that in a minute. And you'll notice it tells you ATG is the initiator base triplet. So it starts all amino acids, but it also codes for methyliodine. And then you'll notice the third note at the bottom is TAA, TAG, and TGA are terminators. So you'll see that at the very end of every protein we make. So how you are going to approach this is I'm going to be giving you guys practice on how to code from non-coding to coding to protein. Non-coding means that we're not going to use it for anything other than to find the complementary DNA. A reminder, complementary DNA is one that matches the strand we have here. Now, complementary doesn't mean identical because that's not. Complementary means that it complements each other. So T's match with A's, and A's match with T's, C's with G's, and G's with C's. That are the only combinations you can do. So when I make my coding strand, I'm making the complementary strand, okay? So T is matched with A is matched with T, C is matched with G, and I do that all the way along. So TTA becomes AAT, triple A becomes triple T, and ATT becomes TAA. So that green strand on your screen now is what we call the coding strand. This strand is what we use to figure out our amino acids. So when we take this strand, we start with ATG, and we remind ourselves that ATG is our start codon. So we would write down start. Then we would go find AAT. AAT is asparagine, and we would do this all the way through. So you would end up with start, asparagine, phenylalanine, and stop. That's how you would write your amino acids. So again, as a reminder, how did I find these? I took these triplets and I coded for them. So I found this is start, this is asparagine, T, T, T. I went T, T, and T, and it was right here, so I wrote it down. T, A is stop. So here's another one. Pause the video, take a moment, and try it yourself. Code from this to your coding strand. So this is our non-coding strand. We're not using it for proteins. It's not coding for proteins. It is just there to make a complementary DNA set. So here's my complementary DNA coding strand. As you can see, A's are matched with T's, and T's are matched with A's, C's and G's, G's and C's. So I have my coding strand there. ATG is my start codon, so I'm going to write down start, and then I'm going to code all the way through. And I end up getting start, glycine, leucine, stop. If you did not get this combination, go back and check why, okay? Start, again, is ATG. G, G, G. Start at G, move over to G, find G. Here's glycine. CTA. C, T, A, leucine. T, A, G, stop. Okay? So that is how you would find and code for different proteins. So what we're going to do is we're going to draw a little quick diagram just to show you essentially what is actually happening after we code. Now, for those of you that may have taken Bio 30 or may understand a little bit more about coding for proteins, there's actually a lot of steps that are involved in this process. It's actually called transcription and translation. And transcription is taking the DNA from inside the nucleus and making sure that we can read it. So essentially going from non-coding to coding and then making something called mRNA. You don't need to know that. But essentially mRNA leaves the nucleus and ends up inside of the cytoplasm, which is outside of the nucleus in the cell. And what it does is it's where we make our proteins using what we would call RNA. But for our purposes in Science 30, you just need to understand the process. So process one is having a coding strand. So in the space that says draw a diagram, I would just recommend drawing something that is a coding strand. This one says TCA, CTT, GGT, and then there's a lone C that we're not going to use. So in the diagram box, I would draw your coding DNA. Then what we're going to do is I'm going to take it and remind myself that I'm taking those triplets and I'm making different types of proteins. So TCA, if you look in your chart, which I'll go back here, TCA right here is serine. So I'm going to make a serine protein, serine amino acid, sorry. I'm going to follow it with a CTT, which is a leucine, and then a GGT, which is a glycine. So again, we're just taking our triplets and coding for some amino acids. Now this amino acid is slowly growing because what's happening is as we make each amino acid, we bond them together with a polypeptide bond. So we're creating a chain of amino acids. Eventually, that chain of amino acids is going to start to fold up to make a protein. And that's what you see here. Is now this amino acid chain is slowly folding itself up to form the correct protein that our body needs. So again, this could be an example of hair color. All right. So your DNA codes for the color of your hair. We make the amino acids in the correct order that causes your hair to be the color it is: brown, blonde, red, black, in an in-between color. And then it folds itself up. This protein folds itself up and then is expressed in your hair color. And that is essentially how we make DNA into a protein. All right. So again, your diagram requires you to just show a coded set of DNA or amino acids and then a folded protein. So make sure you label that: amino acids and folded proteins. So we're going to do lots and lots of practice on this. If you have any questions, please let me know.